Good morning, my name is Christoph Deben and I'm a cancer biologist working as a postdoctoral researcher in the Center for Oncological Research in the University of Antwerp. And together with my teammates Edgar and Abraham, we developed the Orbit's 3D screening platform on which I will give you more information in this presentation. So I've been a cancer researcher for almost 10 years now, and we've seen a clear shift from more general therapies to personalized therapies. And a very interesting development in this field is the ability to grow the tumor of the patient as 3D tissue organoids in the lab. We can do drug screenings on these organoids and then recommend treatments for patients on clinical trial, or at least that's the most ultimate goal of these organoids. So these organoids can really be considered as a patient in the lab and provide a lot of opportunities for preclinical and clinical research. When implementing this technology in the lab, we clearly found two problems. The first one is the lack of sophisticated high-throughput drug screening assays, because the current ones do not provide enough information about the drug mechanism of action. So our solution is based on the development of live cell imaging assays, where we provide more in-depth analysis of the drug response, have the options for kinetic measurements, and still retain a high-throughput format. The second issue we faced was when we tried to analyze our images, we found that there's no user-friendly or sophisticated 3D image analysis available, and our solution was to develop our own AI-dependent image analysis software. This is an example of organoids grown in a high-throughput format microplate, and the software that allows us to track the growth of these organoids, either treated or untreated, and we can also quantify, for example, the amount of cell death that is induced over time. As shown in the previous image, we do not use any labels, so organoid detection is purely based on bright field imaging, which has the benefit that no phototoxicity can be induced, and we fully validate our assays against the gold standard methods like cell-titer glow and fluorescent viability markers, as shown in the graph here, where we show a very strong correlation between our bright field parameters and the gold standard fluorescent parameters. The second advantage is that we have multi-parametric readouts, so we can easily distinguish cytostatic from a cytotoxic response. When growing these organoids in these microplates, we also see that they behave differently. So some are more static, other ones move around more, look for each other, merge together. So these are all parameters we can quantify and use as a parameter when we study new therapies. And we are also in the progress of developing more complex advanced co-cultures to recreate the tumor microenvironment, as shown here, a pancreatic tumor organoid surrounded by cancer-associated fibroblasts. The third advantage is that the analysis is automated, so this can increase the reproducibility, also reduce user-dependent variability, and reduce overall time spent by the researcher on the analysis. So these innovations we want to offer as two independent services. The first one is the drug screening services for universities, biotech, pharma who have a new compound and they want to test the efficacy, but they do not have live cell imaging infrastructure and want to make use of our cancer cell line and organoid biobank. If you have their own cell lines, they can of course send them to us also. So we offer patent-pending multi-parametric drug screening assays where we aim to recreate the tumor microenvironment but still retain a selective readout for each cell type in the co-culture. This is all supported by a state-of-the-art infrastructure, being an open-source pipetting robot, drug printer, and the TCAN Spark Cyto, which is a live cell imaging instrument with also luminescence capabilities and full environmental control. The second service we want to offer is the image analysis software as a web app, where we can also offer cloud data storage, data compression, and also data management, because these live cell experiments can take up a lot of storage. And in addition, we also want to automate the data analysis, so automatic calculation of drug metrics, a graph editor, a video editor. So really an all-in-one solution for people that have their own live cell imaging instrument but want more advanced image analysis. We will also share the in vitro protocols for our assays, provide tips and tricks so you can really implement the drug screening assays we develop in your lab use your own system and use our software to do the analysis. So we also aim to be platform independent. If you have an Inky site, a Spark site, so that doesn't matter. And if you require more advanced analysis, you can also come to us for custom image analysis. So we now received funding to really set up the web app, and it would be very useful to us if you can visit our website and complete our survey to give us more information on what your needs are so that we can implement it in the software. And if you require any other information about the drug screening service or the image analysis service, you can visit the website and contact us on the email shown below. Thank you for listening.
Hello, I'd like to thank the organizers for inviting me to present Juventa Biologicals, a startup project supported by the University of Ghent, the aim of which is to provide minimal invasive treatments to restore sight and prevent incurable blindness. As people get older, part of their proteins become glycated to form AGEs, or advanced glycation end products. AGEs in the serum are biomarkers for AMD and diabetic retinopathy. And in fact, AGEs induce eye disease. In the eye, AGEs cause front of the eye diseases and back of the eye diseases. In the front of the eye, presbyopia and cataract are caused by rigidification and opaqueness of the lens. And glaucoma is linked to elevated intraocular pressure, leading to atrophy of the optic nerve and blindness. In the back of the eye, diabetic retinopathy and age-related macular degeneration lead to blindness. The current therapies only treat the symptoms. There's no cure. The solution that Juventa Biologicals has come up with is to degrade AGEs that can provide a potential game changer for the treatment of ophthalmological diseases. So early intervention can revert the process and restore sight. Juventa Biologicals has two different enzymes, IU1 and IU2, that can take away the sugar from AGEs, reducing the level of AGEs. So Juventa Biologicals tackles the real cause of age-related ophthalmological diseases. And you can see here in the below panel a proof of concept with diabetic OBOB mice, which have been injected in the eye intravitreally, either with control or with IU1. In the control-treated animals, you see the thick inner retina reflecting diabetic retinopathy. In the right-hand panel, with the IU1 treatment, you see normal retina preventing disease prevention and reversion. Over the last few years, Juventa has generated in vitro, ex vivo, and in vivo proof of concepts, both with IU1 and IU2 enzymes, and also with intravitreal injections and with drops. On this slide, we illustrate two different proof of concepts. On the left, human ex vivo donor eyes from stage 3 AMD patients that are treated through in vitro IVT injections with IU1, and you see partial or total clearance of AGEs within a 2-hour period. And in the pictures, you can see a decrease in the volume of drusen, or the OCT lesion area. On the right-hand side, you can see proof of concept in dogs that are blind due to severe cataracts. And here, the eyes were treated with a combination of IU1 and IU2 in the form of drops on the right, or negative control on the left. And you can see almost complete clearance of the cataract, resulting in a reversal of cataract blindness. Also, ophthalmologists can detect early impact on the retina due to age deposition. But there are no treatments for this. This is not reversible. And this can lead to the formation of AMD and blindness. Juventa Biologicals can treat these patients with IU1 or IU2 potentially, leading to reversal of the damage to the retina and preventing blindness. So to look at the back of the eye diseases first, diabetic retinopathy and wet AMD are typically treated by anti-VEGF injections. But this is only effective in some of the patients, and 1/3 of them become anti-VEGF resistant, and you see also induction of atrophy. So a market need will be to intervene early, and being able to treat with drops would be a unique asset. For dry AMD, there's currently no therapy, so it's clear that a treatment that would reverse dry AMD will be desired. For presbyopia and cataract, typically you have surgery in the Western world, but their active retinopathy is a contraindication. So again, eye drops being able to treat presbyopia and cataract would be desirable. The unique selling point of Juventa Biologicals is to have 2 therapeutic leads to treat the cause and allow the early intervention and revert the disease. University of Ghent has supported the generation of a broad IP portfolio covering both know-how but also patents covering IU1 and IU2 therapy in various diseases. For the back of the eye diseases, the size of the addressable market is in each case greater than $1 billion per year, and for dry AMD substantially larger. University of Ghent has supported Juventa Biologicals through industrial research funding and through an AMD chair. And in the near future, Juventa Biologicals will be registered as a company and will seek Series A financing. We should obtain this by Q2, Q3 of next year, with an aim to achieve value inflection by 2025. The deliverables are firstly to prioritize a first therapeutic, secondly to generate a GMP batch and perform tox study, and then file an IND on the first indication for the first therapy. And the first milestone, the first inflection point, will be the completion of phase 1 clinical trial. Exit strategy will be a partner or a trade sale or an IPO after reaching the value inflection point. The spin-off team is already in place. Myself, Elizabeth Van Aken, an ophthalmological surgeon and the head of the retina clinic, SES, Professor Joris de Lange from the University of Ghent, who has a wide experience in glycation. Also supported by Nico Calawart from VIB for enzyme production, and Daisy Flames from the University of Ghent as a business developer. So to conclude, the unique selling point of Juventa Biologicals is to tackle the real problem, allowing early intervention, restoring sight, and prevent blindness, and by that, to substantially improve the quality of life of patients. Thank you very much.
Hello everyone, my name is Maya Inbrechts and I'm working as a postdoc and project manager at Pharmaps, the KU Leuven Antibody Center. Today I'm going to present to you MapMine, which is an integrated platform for the identification, isolation and characterization of human monoclonal antibodies. The MapMine platform is being developed by Pharmaps together with MeBio's Biosensors group from the KU Leuven. So monoclonal antibodies are proteins expressed by B cells that identify or neutralize a target, such as a pathogen or foreign agents, with a high specificity and low toxicity. And this makes them very attractive therapeutic agents. Therefore, monoclonal antibodies are already being used as a therapy for various diseases, such as infectious diseases, cancers, central nervous system diseases, autoimmune and cardiovascular diseases. And over 100 monoclonal antibodies are already approved as a treatment in Europe or in the United States. However, major challenges remain in the antibody discovery process. And one of the first challenges here is to achieve a high throughput screening of B cells in order to get a large and diverse monoclonal antibody panel. And in fact, this is one of the limitations of one of the current available technologies, hybridoma technology. In addition, in order to select the most optimal antibodies, it's most interesting to go for the in vivo matured antibodies because these have a maximal efficacy and quality. And of course, it's also the aim to complete the antibody discovery process as fast as possible. And therefore, we've come up with a solution to address these challenges with our MapMine platform. And here we start from human subjects to isolate PBMCs and the B cells of interest that express a specific antibody. These B cells are then isolated on an individual level and are used to sequence the antibody variable domains. And with this sequence, then the antibodies can be made in vitro recombinantly. And by starting from human subjects, we immediately go for the most optimal antibodies that are in vivo matured, and we also omit the need for humanization afterwards. The throughput of the process is determined by the ability to screen different B cells and to select the most optimal ones. And here are a number of options. One of the most commonly known options is fluorescent activated cell sorting, or FACS. But recently also some other technologies emerged that use either droplets or microarray structures to trap and interrogate single B cells. Several devices are available to perform FACS, but also for the more recent technologies, there are some commercial options. And in fact, we have FACS available at KU Leuven, but we'll also evaluate, and if a positive evaluation, purchase the other technologies at our facility. In addition, we're also working on our own platform, MicroMapMine, to allow for full flexibility of B cell screening and characterization before the cell isolation. And for our first proof of concept study, we used FACS to isolate anti-SARS-CoV-2 specific antibodies from COVID patients. And in this proof of concept study, we started from PBMCs of convalescent COVID patients, and we selected the antigen-specific B cells by using fluorescently labeled receptor binding domain or full-length spike antigens. We were able to isolate more than 1,000 SARS-CoV-2 specific B cells, and of 300 of these, we amplified the antibody variable domains, and we ended up with 67 paired IgG and 15 paired IgM antibody sequences. And of these, we started with the production of 20 antibodies in vitro. And when we characterized these antibodies afterwards, we were happy to see that they all retained their antigen specificity after production, and even bound their antigen with high affinities and very low KD values of below 0.5 nanomolar. 8 of the antibodies were also able to neutralize the virus in vitro with high potency and IC50 values below 1 nanomolar, not only for the Wuhan virus, but also for the alpha, beta, gamma, and delta variants. And 4 of the antibodies were evaluated for their treatment efficacy in an in vivo hamster model, and we could show that one of our antibodies was actually among the best in class when compared to the commercially available ones, and could treat COVID symptoms in hamsters with a high efficacy and a very low dosing. So to summarize, with MapMine, we provide a platform for antibody discovery and characterization in order to bridge the valorization gap with either academia or research centers that often find interesting therapeutic targets, and on the other hand, the industry that is actively looking for new antibody lead candidates and an accompanying data package to strengthen their pipeline. As always, this platform is a team effort. We have an innovation manager and 2 postdocs working on this project together with technical support and 3 academic advisors. Of course, I'm very happy to take your questions, either now or don't hesitate to reach out via email afterwards. Thank you.
Good morning. In the next couple of minutes, I would like to introduce the core facility for microfabrication of microfluidics in glass and silicon, a new core facility of Free University of Brussels, VUB. Microfluidics is an area that sees tremendous growth in the last couple of years. And we see that from increased interest from industry and research groups in and outside VUB, and that's not a surprise because in Belgium, in Brussels, in Flanders, we have quite a bit of a hotspot in this field, of medical, pharma and biotech applications. And it's also one of the key enabling technologies recognized by Europe. And this growth is reflected in the figures we can see in the market. This is, for instance, a recent study showing that in microfluidic applications in the last couple of years and in the coming years, we expect more than 20% growth, specifically in vitro diagnostics, life sciences and related areas. And it's with that background that VUB decided to start a new core facility, more specifically build a new open access clean room that will specialize in the deep reactive ion etching of glass next to silicon, where we have already many years of expertise built. But glass is a material in microfluidics with all kind of particular positive characteristics which we want to exploit further. And we want to build around this material a unique expertise in Belgium and surroundings. This core facility will be called Microlab, and it's not just to serve the microfluidic community of VUB. It's there to serve many different research groups in VUB, in other institutions, and especially also the many small and larger companies surrounding us in this field. And the multidisciplinary aspect of microfluidics is a kind of reflected in a number of projects which I will not discuss in detail, but it gives you already an idea in how many directions we can go. We have at VUB a dedicated microfluidics research group, microflow group, since almost 10 years. But many of the research is not specifically on the microfluidic device, more there to support other fields. I give an example. Biocondensates is to study new developments against Alzheimer. EasyChem is in continuous separation. It's for the chemical industry. Colores is more industrially oriented, multi-layer coatings or optically active coatings of very small particles. And it goes in a very wide scale, always multidisciplinary type of research or research with companies, small biotech companies up to large pharma companies. And an interesting field maybe there to mention, bringing it all together, is the acoustofluidics, a field in which we specialized at VUB since several years. And this is using acoustics to manipulate particles in flow in a microfluidic or in a microscopic scale. And this is small in design, but the use is there for the pharmaceutical industry, for the biotech industry. And this is a typical example where we want to bring acoustics onto a liquid medium, which requires the use of hard and stiff materials such as glass or silicone. So we will build a 550 square meter clean room right in the middle of the Etterbeek campus in one of these old student houses, Willy van der Mere, which are protected. So it's a very nice area. It will be accessible with wheelchair, with offices for users, and we plan a capacity of 5 to 8 full-time equivalent users. We expect to be operational end of 2022, and the works are starting as we speak here. So a brand new clean room, 300 square meter class 1 with 250 square meter technical areas. And we will bring, of course, to master glass cutting-edge technology, more specifically deep reactive ion etching of glass, of silicon, and all the supporting technologies like lithography, thin film deposition, plasma, and so further. To stress, and maybe to conclude, it's going to be an open access facility, and that's why we're happy to launch it for the first time here at Knowledge for Growth, because it's not there just for the VUB community. It's not there just for the academic community. It's there for the companies surrounding us in our biotech ecosystem. And the vision we have is that we will have internal users, external users, and it's against a usage fee that people or researchers, and after training, of course, will be able to access this clean room and use the state-of-the-art equipment. A preliminary pricing schedule is shown here, would be €135 per hour of clean room, and these are very attractive rates if we compare, for instance, with a Mesa Plus or a DTU here in Europe. This whole project is quite ambitious for us, and we're very happy that VUB as a university has provided significant funding to realize this project, but also to mention that we got significant support from the Vlaamse Veerkracht Relance Plan that was launched some time ago. Please be invited to connect with me, learn more about it, and maybe plan for future activities, collaborative research that can use this type of technology.
Hello, my name is Emiliano Mancini, and I'm a business developer at the Data Science Institute of UU Hasselt. The Data Science Institute is one of the 4 spearhead institutes of UU Hasselt, and it's located in Campus Diepenbeck. It was founded in 2020 by pooling the scientific excellence of UU Hasselt researchers active in the field of data science and working on different research groups focusing on natural language processing, statistics, computational mathematics, computer science, and the integration of clinical data. The Data Science Institute aims to bring together high-quality, data-driven research, education, and industrial collaboration, both at local and global level, to facilitate a data-driven future. Data Science Institute aims to act as a central point of contact for data science, leveraging the different synergies between data science domains. In other words, we aim to be a one-stop shop for data science. For this reason, we focus on the whole data science cycle. We develop methods to understand, visualize, and quantify user context and user needs, methods for automated data retrieval, data management, and data wrangling, as well as methods for high-dimensional data exploration. Of course, we also develop models such as statistical models, machine learning, AI, and mathematical models, as well as their analysis. We also develop methods to evaluate such models, for example, assessing their speed, accuracy, explainability, and replicability. Finally, we develop proof of concept software and tools that can be further implemented by users. For this reason, we work on different application domains, and the tools that we develop for some of these application domains can be transferred to other application domains. For this reason, we are open to explore new opportunities in different applications. We are active in different, as you said, in different application domains, and here are some of them. As you can see, we are very active in the life sciences, working on infectious diseases in humans and animals, including the COVID-19 pandemic. We work on drug discovery and the discovery of biomarkers, as well as design and analysis of clinical trials. We have experience in microbiome, mass spectrometry, proteomics, and tumor heterogeneity. Because of this, we are quite experienced in working with the health data, and we can ensure to work on this data in a secure way, ensuring GDPR compliance. Of course, we also work on other domains. For example, business intelligence, administration and logistics, decision support system. We also work on multi-objective optimization, mobility. We have experience in flow modeling, food safety, but also in environmental research and even archaeology. We are open to collaborate in new applications, and these are some of the problems that we can solve at the Data Science Institute. We can work on visualization and exploration of complex data, support an efficient data governance, as well as a correct and fast querying of databases. We can develop decision support system and multi-objective optimization. We are able to integrate data from different sources in a comprehensive way and avoid bias when processing big data. We can analyze big but imperfect data, detect and predict risk or anomalies. We have experience in designing and analyzing clinical trials, also with limited cohorts, but also other problems such as software optimization, contextualization of data, and semantics. So if you have problems in some of these fields, you are welcome to contact us. So how to collaborate with us? We have 3 different channels: consultancy, student projects, and joint projects that can be either PhD projects or multi-stakeholder research projects. For consultancy, we usually start with an intake meeting to define the challenges and match these challenges with the available expertise at DSI. We have extensive experience in consultancy, and you are welcome to explore this opportunity. However, we also work on student projects. We have 3 different types of collaborations for these. One is Study Week Mathematics for Industry in the context of the Bachelor of Mathematics. But we also have thesis projects that are usually handled in collaboration with an external supervisor and the supervisor from the Data Science Institute in the context of the Master of Statistics and Data Science and the Master in Computer Sciences. Finally, we work on joint projects. For PhD projects, we work either on fully sponsored fully sponsored PhD projects, fully paid by the external organization, or co-funded by the UHasselt Civic PhD program. For research projects, we again work either on fully sponsored projects by the external organization, or we can apply together with the external organization to funded or co-funded projects, usually in collaboration with FLYO and FWO. So we look forward to collaborate with you. If you have questions, you are welcome to ask after this talk. But you can also reach out to us via email, either to Sarah Vercruis, my colleague, or myself. Thank you for your attention.